Welcome back to Antibody Basics by Biointron, Part 14. In this episode, we'll talk about therapeutic targets, inflammatory diseases. Antibody-based therapeutics are engineered antibodies that target specific molecules in disease, such as in autoimmune diseases, cancer, and infectious diseases. They are important because they can be highly specific, have a long half-life, and multiple mechanisms such as neutralization, immune modulation, and cell depletion. Chronic inflammatory diseases are mediated by dysregulated immune responses involving cytokines, immune cells, and tissue damage. Antibody therapies provide targeted intervention, reducing inflammation without broad immunosuppression. For example, TNF-alpha inhibitors like infliximab and adalimumab for rheumatoid arthritis and IBD, as well as IL-6 inhibitors such as tocilizumab for autoimmune diseases. Antibodies have several functional properties, with the antigen binding in the Fab region recognizing and binding to specific epitopes on target molecules, as well as determining specificity of monoclonal antibodies. Effector functions include antibody-dependent cellular cytotoxicity, or ADCC, and complement-dependent cytotoxicity, or CDC. Therapeutic monoclonal antibodies are engineered for a longer half-life and optimized binding affinity. Neutralizing antibodies are used for therapeutic purposes to block cytokines, for instance, anti-TNF for inflammation, depleting antibodies for targeting immune cells such as anti-CD20 for B-cell depletion, and immune checkpoint inhibitors to block inhibitory receptors such as PD-1 and CTLA-4. Inflammation is a protective response by the immune system to injury, infection, or toxins. It involves immune cells, blood vessels, and molecular mediators. Anti-TNF therapies, such as infliximab and adalimumab, block TNF-alpha, thus reducing inflammation in rheumatoid arthritis, IBD, and psoriasis. IL-6 inhibitors, such as tocilizumab, inhibit cytokine signaling, thus decreasing chronic inflammation. B-cell depletion, such as with rituximab, removes autoreactive B cells, thus treating lupus and rheumatoid arthritis. With inflammatory diseases, monoclonal antibodies work in several ways. For cytokine neutralization, antibodies can bind to TNF-alpha, IL-6, or IL-17, preventing inflammation. With receptor blockade, antibodies can block IL-6R, IL-23R, stopping immune activation. With cell depletion, anti-CD20, such as rituximab, eliminate B cells in autoimmune diseases. Immune checkpoint modulation, such as with anti-PD-1 nivolumab, enhances immune regulation. There are several therapeutic implications. First, selective targeting reduces off-target effects compared to steroids. Second, long half-life, which means fewer doses needed for sustained action. And third, potential for combination therapy, thus targeting multiple pathways for better disease control. These therapies are highly specific, targeting disease pathways with fewer side effects than steroids, have a longer half-life, thus requiring less frequent dosing than traditional treatments, and proven efficacy, used in millions of patients worldwide. However, there are challenges and limitations. For instance, risk of infections due to immune suppression, high cost compared to small molecule drugs, and potential loss of efficacy due to the formation of anti-drug antibodies. Here are a few examples of therapeutics. Enbrel is a TNF receptor Ig fusion protein, which binds to TNF-alpha to block its activity. Remicade is a TNF-neutralizing monoclonal antibody, which binds to TNF-alpha, both soluble and membrane-bound. Tocilizumab is an IL-6R neutralizing monoclonal antibody which blocks IL-6R to inhibit signaling. Cosentyx is an IL-17A neutralizing monoclonal antibody which neutralizes IL-17A cytokine. Ustekinumab is an IL-12 and IL-23 neutralizing monoclonal antibody which blocks that pathway. Biointron focuses on antibody discovery, expression, and optimization. From gene sequence to purified antibodies, it takes just 2 weeks. Biointron's high-throughput platform enables the rapid expression of recombinant antibodies in HEK293 or CHO cells. We guarantee the delivery of 100 micrograms to 100 grams of recombinant antibodies. Biointron is also a well-recognized leader in VHH antibody discovery and production. With our self-owned alpaca breeding farm, we offer a 1 alpaca for 1 project commitment and guarantee the delivery of multiple binders, high diversity, and large capacity. Biointron also has several other services, including but not limited to bispecific antibody production, where most formats can be produced, with thousands of bispecific antibodies already successfully delivered. Afucosylated antibody production, as afucosylation has been shown to significantly boost ADCC effects. Single B-cell screening, with 2 to the 10 by the power of 6 plasma B cells from immunized animals, hybridoma sequencing, cross-verified with 5 clones and 100% sequencing accuracy guaranteed, and both antibody humanization and affinity maturation for antibody optimization, as well as recombinant protein expression in the mammalian system, and a high yield guaranteed with our ECA-CC licensed CHOK1 stable cell line. Thank you for watching, and comment with any questions or topics you'd like us to cover next. Don't forget to like and subscribe for more.